In this video, we will discuss about how this UCP1 CRISPR adipose organoid suppress cancer progressions. So in the previous video, we have understood that these adipocytes can suppress tumors in this xenograft models. Here what we did is, here what the researchers did is they co-transplant this organoid, that is UCP1 CRISPR adipose organoids, along with cancer. At the same time, they feed the mice with different food and then check whether what are the changes it does to the tumors. So these things, the results of that, we will discuss it today. But before that, we will discuss some of the markers that gets affected when this UCPR1 CRISPR adipose organoids co-transplanted with the tumors, and for the control they use VP94 CRISPR adipose co-transplanted with the cancer. And VP94 does not have too much nutrient intakes, or that gene's expressions does not cause too much nutrients intake by the adipose, so that acts as a negative control. Here when compared to these adipose organoids, that is for the control, when they use VP 94 or 64, you can check that from the paper. They see that when they use UCP1, the this luminescence per mg of tissues, or we can say the consumptions of these nutrients will be higher when this UCP1 adipose is used. And when they compare it with the tumors co-transplanted with this UCP1 CRISPR adipose organoids, they see that when in the tumors the amount of nutrients goes down when they use this UCP1, when the tumors are co-transplanted with this UCP1 CRISPR adipose organoid, indicating that the UCP1 somehow disrupts this tumors nutrient supplies. And when they check some of the other factors such as glucose 6-phosphate, fructose 6-phosphate, 3-PG, PEP, they see that its relative abundance goes down in the case of these tumors when they are co-transplanted with this UCP1 CRISPR adipose compared to the control. And in terms of the fatty acid, they check for the oleic acid and palmitoleic acids. They see that this signal also goes down when the tumors are co-transplanted with this adipose, UCP1 CRISPR adipose, compared to the control where they use VP94. So all this indicates that when the tumors are co-transplanted with this adipose organoid, it it disrupts their nutrient supplies. So what they did is they designed the experiments where they did a similar thing, that is xenograft cancer cells. After 6 weeks, they transplant this CRISPR adipose organoid with UCP1 gene with UCP1 gene transfected, and for the control VP94 genes is transfected. And then they feed the mice with 3 different food, that is CHO, HFD, the high fatty acid diet, and 15% glucose diet, and then check the tumors for this tumor growth, volume, metabolic genes, hypoxia, angiogenesis. And when they use the CHO diet, which is the regular diet, they see that decrease in the tumor volume when they use, then the tumors are co-transplanted with this UCP1 CRISPR adipose organoid compared to the control. But for this high fatty acid diet and 15% glucose diet, they do not see any change in the tumor weights. And these things they are quantified on the right hand side, where we can see that for the CHO diet the tumor size goes down, but for the high fat diet or 50% glucose diet there is no change, indicating that the UCP1 gene might disrupt this high fatty acid and might disrupt this fat consumptions, or might compete with the tumors for the fat consumptions and the glucose. And when they are used in excess amount, there is enough fats and glucose available for both the adipose and the tumors. And they check the other markers also for the proliferation markers. They can see the proliferation markers also goes down for the CHO diet but not for the other diets. And the other markers such as GLUT4, GCK, CD36, NCP, T1B also shows significant changes for the CHO diet but not for the other two conditions. So this all substantiate their finding the previous hypothesis that CHO diet might CHO, this UCP1 adipose organoid might compete the high might compete with the fat consumptions and the glucose fat consumptions and the glucose consumptions. So in order to further substantiate that, what it is they check, and this MIK67 is not the proliferation markers. These are the markers for the this these are the markers for the tumor progressions, we can say that. And the proliferation markers is Ki67. So the Ki67 also goes down. And for this CA9, which is the markers for hypoxia, it also goes down in the CHO. And the CD31, which is the marker for microvessel density or angiogenesis, we can say that it also goes down for the CHO but not for the other conditions. So these results are also inconsistent with their cancer proliferations markers and this tumor size data. Now they check these differential gene expressions, and in this case what they are able to find out that in the case of CHO there is a differentially regulated genes they have observed. That is 6623 genes are down regulated and 479 genes are up regulated, but there is not so much change they have observed for HFD and glucose compared to its control. And in terms of this upregulated genes, what they are able to find out that upregulated genes are mainly related to the cancer suppressions. The downregulated genes are mainly related to the cancer progressions. So all this data finally made a point or support their hypothesis that this UCP1 CRISPR adipose organoid might compete for the fat and glucose consumptions with the cancer cells. And when these fat and glucose are in high amount in their diet, they are able to suppress the properties. They are not able to, this adipose is not able to suppress the cancer progressions, as in the high fat and glucose diets the amount of fat and glucose is enough for both of them. So that's all for this video. If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.